Hello everyone and welcome to this educational series on biochemistry and clinical pathology for the pharmacy students. In today's video, we are going to start with the second chapter having name carbohydrates. This chapter having 6 marks. So these are the syllabus points of the chapters. First is definition, classification with example and chemical properties of the carbohydrate. The second point is the monosaccharide. Monosaccharide actually a class of the carbohydrates. In this, we will study structure of glucose, fructose and galactose. Then next is the disaccharide. In this, we study structure of maltose, lactose and sucrose. Then next is polysaccharide. In polysaccharide, we study chemical nature of starch and glycogen. Then next and last point of the syllabus is quality test and biological role of the carbohydrates. So first, we start with the definition of the carbohydrates. Carbohydrates are polyhydroxyaldehyde or ketones or compounds having general formula CnH2nOn. Then the classification of the carbohydrates. So two major classes of carbohydrates are sugar and non-sugar. Sugar type of carbohydrates are classified into two groups based on the number of the sugar units. So in sugar part, two parts are divided. First is monosaccharide and next is oligosaccharide. The third part of this classification is the non-sugar, which is a polysaccharide. So first we start with monosaccharide. Monosaccharide are simplest group of carbohydrates and often referred to as a simple sugar. They have general formula CnH2nOn and they cannot be further hydrolyzed. They are further classified as two groups. First is on the basis of functional group and second is on the basis of number of carbon atoms. In this functional group, first class is aldolates and second class is ketones. When the functional group of monosaccharide is an aldehyde, then they are known as the aldolates. And second class is ketones. When the functional group of monosaccharide is ketone, then it is known as a ketones. These are the only sub difference, that is monosaccharide having functional group aldehyde, then it is known as a aldolase, and monosaccharide having functional group ketone, then it is known as a ketones. These are the examples of the aldolase and the ketones. Then second part on the basis of number of carbon atoms. So this is a simple part. First is trioses, 3 carbon atom known as trioses. Examples are the glyceraldehyde and dihydroxyacetone. Then second is tetroses, 4 carbon atom are known as a tetroses. Example erythros and erythrolones. Then next is pentoses, 5 carbon atoms. Example ribose and ribulose. Then next is hexoses, 6 carbon atom. Example glucose, fructose, galactose and mannose. Then next is heptoses, 7 carbon atoms. Example pseudoheptoses and pseudoheptuloses. Then next is after monosaccharide, the next class is oligosaccharide. Oligosaccharide contain 2 to 10 monosaccharide molecules. Based on the number of monosaccharide unit present, the oligosaccharide are further subdivided into disaccharide and trisaccharide. So first class is monosaccharide. Monosaccharide having further classified as on the basis of functional group and on the basis of number of carbon atoms. Then the second class is oligosaccharide. In oligosaccharide, subtypes are disaccharide and trisaccharides. So disaccharides are class of oligosaccharide containing 2 monosaccharide units. Disaccharides contain 2 monosaccharide units and trisaccharide contain 3 monosaccharide units. Then third part or third class of the carbohydrate is non-sugar, which is also called as a polysaccharide. Polysaccharide are polymer of monosaccharide units with higher molecular weight held together by the glycosidic bonds. Polysaccharide again having two types. First is homopolysaccharide and the second is heteropolysaccharides. Homo means same, hetero means different. These on the hydrolysis yield only single type of monosaccharide unit known as a homopolysaccharide. Example are starch, cellulose and glycogen. Then next is heteropolysaccharide. These on hydrolysis yield a mixture of few monosaccharide or their derivatives unit. Example hyaluronic acid, heparin and clondropin sulfate. This is the summarization of classification of carbohydrates. Carbohydrates are classified into monosaccharide, oligosaccharide and polysaccharide or also called as a non-sugar part. So, second point of the syllabus is monosaccharide. In monosaccharide, we study structure of glucose, fructose and galactose. This is the structure of glucose, fructose and galactose. Then next is the disaccharide. This is the structure of the maltose, sucrose and lactose. Maltose, sucrose and lactose. Then next is the polysaccharide. In polysaccharide, we will study chemical nature of starch and the glycogen. So first is the starch. Starch is a polysaccharide. Starch is the most important source of carbohydrate in human diet. Starch is a homopolysaccharide of D-glucose. Starch is the mixture of two polymers, amylose and amylopectin. So, first polymer is the amylose. Amylose is a linear polysaccharide composed of entirely of D-glucose units. This is the structure of the amylose. Then next is the amylopectin. Amylopectin is a branched polysaccharide composed of glucose unit. So this is the structure or chemical nature of the starch. Starch, which is a mixture of two polymer, amylose and amylopectin. Then next is glycogen. This is a structure of amylopectin. Then next is a glycogen. It is reserved carbohydrate. This is polysaccharide made up of glucose units by 1,4-glycosidic linkage. It is non-reducing sugar and soluble in the water. This is the structure of glycogen molecule. Then next point of this syllabus is qualitative test of carbohydrates. So first test is Mollis test. In Mollis test having principle, carbohydrates are dehydrated by concentrated sulfuric acid to form purpuril which is react with alpha naphthol to give a violet ring. Then this is the inference of Mollis test. A violet ring at the junction indicates the presence of carbohydrates. The next test is Benedict's test. In Benedict's test, the principle is reducing sugar, reduce copper sulfate in Benedict's reagent to form a red or orange color. Then inference is formation of a red or orange precipitate indicates reducing sugar. The next test is Barfor's test. In that, principle is distinguish monosaccharide from disaccharide using copper acetate in acidic medium. Then next is inference. In inference, a red precipitate formed within 2 minutes indicates a monosaccharide. Then next point or the last point of this syllabus is biological role of carbohydrates. First is Carbohydrates are main source of energy. They are essential component of some structural materials in living organisms. They are necessary for lipid metabolism. Heparin prevents blood coagulation. Heparin is a carbohydrate or example of carbohydrate. Hyaluronic acid acts as a lubricant. Then carbohydrates prevent constipation. Then the previously asked questions are draw the structure of any three reducing monosaccharide. So these are the monosaccharide glucose, fructose and galactose. So the structure is per structure 1 mark. Then next is define carbohydrate and classify them. Then next is what are carbohydrates? Classify them with suitable example. And next question is the nitrogen base found in RNA but not in DNA is. Thank you.